CNA Handbook Review. Welcome to the quiz. Question 1. Parkinson's disease involves degeneration of a specific dopaminergic pathway. Which nuclei in the midbrain are the primary source of these neurons, and what is the major clinical consequence of their loss on basal ganglia circuitry? Option A. Locus coeruleus norepinephrine results in intention tremor and ataxia. Option B. Raphe nuclei serotonin results in depression and sleep disturbances. Option C. Substantia nigra pars compacta results in increased inhibition of the thalamus and bradykinesia/rigidity. Option D. Ventral tegmental area results in a flattening of effect and anhedonia. The answer is C. Parkinson's disease is characterized by the progressive loss of dopamine-producing neurons in a specific region of the midbrain called the substantia nigra pars compacta SNC. These neurons project their axons to a part of the basal ganglia called the striatum caudate and putamen, forming the nigrostriatal pathway. In the normal basal ganglia circuit, dopamine from the SNC facilitates the direct pathway, which promotes movement, and inhibits the indirect pathway, which suppresses movement. The net effect is to allow desired movements to proceed. When dopamine is lost, the indirect pathway becomes overactive, and the direct pathway becomes underactive. This leads to excessive inhibitory output from the basal ganglia, specifically from the internal segment of the globus pallidus, GPI, to the thalamus. With the thalamus excessively inhibited, it cannot properly activate the motor cortex, resulting in the classic motor symptoms of Parkinson's: bradykinesia, slowness of movement, rigidity, resting tremor, and postural instability. Option A describes the locus coeruleus, which produces norepinephrine. Its degeneration is associated with conditions like Alzheimer's, but not primarily Parkinson's motor symptoms. Option B describes the raphe nuclei, which produce serotonin. Option D describes the ventral tegmental area VTA, which sends dopamine to limbic areas and is involved in reward and motivation. Its dysfunction is more linked to psychiatric symptoms, not Parkinson's primary motor deficits. Question 2. Cerebrospinal fluid CSF has several key functions. Where is the majority of CSF produced, and through what structures is it primarily reabsorbed into the venous system? Option A. Produced by the ependymal cells lining the ventricles, reabsorbed via the choroid plexus. Option B. Produced by filtration from cerebral capillaries, reabsorbed into lymphatics. Option C. Produced by the choroid plexus in the ventricles, reabsorbed via arachnoid granulations into the dural venous sinuses. Option D. Produced in the subarachnoid space, reabsorbed via the foramen of Magendie. The answer is C. Cerebrospinal fluid is a clear fluid that cushions the brain and spinal cord. The majority of CSF, about 80%, is actively produced by specialized structures called the choroid plexus. The choroid plexus is a network of blood vessels and ependymal cells located within the ventricle's hollow spaces of the brain. It filters blood plasma and secretes CSF. CSF flows from the ventricles into the subarachnoid space surrounding the brain and spinal cord. It is not reabsorbed by the choroid plexus. Instead, CSF is primarily reabsorbed back into the bloodstream through structures called arachnoid granulations or villi. These are projections of the arachnoid mater, the middle meningeal layer that protrude into the dural venous sinuses, which are large blood-filled channels. The pressure of the CSF pushes it through one-way valves in these granulations and into the venous blood. Option A is incorrect because while ependymal cells line the ventricles, they are not the primary producers. The choroid plexus, which includes modified ependymal cells, is. Also, the choroid plexus produces, not reabsorbs, CSF. Option B is incorrect. CSF is not a simple filtrate from general capillaries, and lymphatic reabsorption is minor in the CNS. Option D is incorrect. The foramen of Magendie is an opening that allows CSF to flow out of the fourth ventricle into the subarachnoid space, not a site of reabsorption. Question 3. The stretch reflex, e.g., patellar reflex, is a monosynaptic reflex arc. What is the name and location of the sensory receptor that initiates this reflex when the muscle is suddenly lengthened? Option A, Golgi tendon organ, located in series with muscle tendons. Option B, muscle spindle, located in parallel with extrafusal muscle fibers. Option C, Pacinian corpuscle, located in subcutaneous tissue. Option D, nociceptor, located in skin, muscle, and viscera. The answer is B. The stretch reflex is the body's automatic response to a sudden stretch of a muscle, like when a doctor taps your patellar tendon. The sensory receptor that detects this stretch is called the muscle spindle. Muscle spindles are specialized sensory organs embedded within the belly of the muscle itself. They're arranged in parallel with the main contractile muscle fibers, extrafusal fibers. This means when the whole muscle is stretched, the muscle spindle is also stretched. The spindle contains intrafusal muscle fibers and sensory nerve endings that wrap around them. When stretched, these sensory endings fire signals. The signal travels very fast via a sensory afferent neuron directly to the spinal cord. In the spinal cord, this sensory neuron makes a single, direct monosynaptic connection with a motor efferent neuron that goes back to the same muscle. This motor neuron then causes the muscle to contract, counteracting the stretch. Option A, the Golgi tendon organ, is located in the tendon in series with the muscle and senses tension, not length. It triggers the inverse stretch reflex, causing relaxation to prevent muscle damage. Option C, Pacinian corpuscles, detect deep pressure and vibration. Option D, nociceptors, detect painful stimuli and trigger withdrawal reflexes, which are polysynaptic and more complex. Question 4. The autonomic nervous system has two major divisions. Which division is characterized by long preganglionic and short postganglionic neurons and uses acetylcholine as its primary neurotransmitter at both ganglia and effector organs? Option A, the somatic motor system. It innervates skeletal muscle. Option B, the sympathetic division. It uses norepinephrine at most effectors. Option C, the enteric nervous system. It is intrinsic to the gut. Option D, the parasympathetic division. It is rest and digest. The answer is D. The autonomic nervous system ANS controls involuntary functions like heart rate and digestion. It has two main branches with opposite effects. The parasympathetic division is responsible for rest and digest activities, conserving energy. Its anatomical hallmark is long preganglionic neurons that originate in the brainstem or sacral spinal cord and travel a long distance to ganglia located very near or within the target organs. Because the ganglia are close, the postganglionic neurons are very short. Both the preganglionic neuron at the ganglion and the postganglionic neuron at the effector organ release the neurotransmitter acetylcholine ACH. This is in contrast to the sympathetic division. Option A is incorrect. The somatic motor system controls voluntary skeletal muscles and uses a single neuron, not a two-neuron chain that releases ACH at the neuromuscular junction. Option B is incorrect. The sympathetic division has short preganglionic and long postganglionic neurons and uses ACH at the ganglion, but norepinephrine at most effector organs except sweat glands. Option C, the enteric nervous system, is a complex network within the GI tract wall and is considered a separate division of the ANS, but it is not characterized by the long pre/short postganglionic structure of the parasympathetic outflow. Question 5. Hearing relies on the transduction of sound waves into neural signals. In the organ of Corti, what is the specific mechanism by which stereocilia bending leads to depolarization of the hair cell? Option A, the bending opens voltage-gated sodium channels. Option B, the bending opens mechanically gated potassium channels, allowing K influx from the endolymph. Option C, the bending closes chloride channels, leading to a more negative potential. Option D, the bending triggers the release of glutamate from the base of the cell. The answer is B. Sound waves cause vibrations in the fluid-filled cochlea, which moves the basilar membrane. Sitting on this membrane is the organ of Corti, which contains the sensory hair cells. Each hair cell has a bundle of stiff hairs called stereocilia on top. The tips of shorter stereocilia are connected to the sides of taller ones by tiny filamentous tip links. When sound vibrations move the basilar membrane up and down, the stereocilia bundle bends. Bending in the direction of the tallest stereocilia stretches the tip links. This stretching pulls open mechanically gated ion channels at the tips of the stereocilia. The unique feature here is that the fluid surrounding the stereocilia endolymph is unusually rich in potassium K. Therefore, when these channels open, it is primarily potassium ions K that rush into the hair cell down their electrochemical gradient. This influx of positive ions depolarizes the hair cell, makes it less negative inside. This depolarization then opens voltage-gated calcium channels at the base of the cell, leading to neurotransmitter glutamate release. Option A is incorrect because the initial channels opened are mechanically gated, not voltage-gated. Option C is incorrect. Depolarization is caused by an influx of positive ions, not by closing channels. Option D describes a consequence of depolarization neurotransmitter release, not the initial transduction mechanism. Question 6. Neurotransmitter action must be terminated. At the cholinergic synapse, what is the enzyme that rapidly hydrolyzes acetylcholine into inactive metabolites, and where is it located? Option A. Catechol-O-methyltransferase COMT, located in the synaptic cleft. Option B. Monoamine oxidase MO, located in the presynaptic mitochondrial membrane. Option C. Acetylcholinesterase ACHE, located on the postsynaptic membrane. Option D. Glutamate decarboxylase, located in the cytoplasm of inhibitory neurons. The answer is C. For a synapse to function properly, the signal must be turned off quickly after it is sent. At synapses that use acetylcholine ACH as a neurotransmitter, cholinergic synapses, like the neuromuscular junction, the primary mechanism for terminating the signal is enzymatic breakdown. The specific enzyme is acetylcholinesterase ACHE. ACHE is anchored into the postsynaptic membrane, with its active site facing the synaptic cleft. When ACH is released and binds to its receptors, ACHE is right there to break it down. It hydrolyzes ACH very rapidly in milliseconds into two inactive products, choline and acetate. This rapid breakdown ensures that ACH does not continue to stimulate the postsynaptic cell, allowing for precise control of muscle contraction or nerve signals. Option A, COMT, breaks down catecholamines like dopamine and norepinephrine. Option B, MAO, also breaks down catecholamines and serotonin inside neurons. Option D, glutamate decarboxylase, is the enzyme that makes the inhibitory neurotransmitter GABA from glutamate. Great job! Don't forget to subscribe. Bye!